So this is basically how the outer ear works. Now, something that's kind of interesting, I mentioned that depending on the type of animal you are, particularly the type of the mammal you are, your ears are probably going to look a little bit different. So what you're looking at here are different types of mammals, and you can see that pinna are very, very different across mammals. So here we have a human ear. I believe this is a capybara. This is a seal. Look at those cute little ears. Here we have a rabbit or a hare, a monkey, an elephant, and a very cute bat. Well, I think bats are cute anyway. So pinna are different depending on the particular needs of that given animal. So now we're going to go into the middle ear. So I mentioned that sounds are funneled in by the pinna, they travel down the ear canal, and they vibrate against the eardrum. The middle ear starts right after the eardrum. So you can actually see these 3 different tiny bones that are attached to the back of the eardrum. These are what are known as the ossicles. So the ossicles are connected to the eardrum and the cochlea. So we have the malleus, which is the hammer, that is attached to the eardrum. We have the incus, or the anvil, that is the middle. And then we have our stirrup, or stapes. You can see it actually looks like a stirrup. And that is attached directly to the oval window of the cochlea at its base. So one of the things that we'll see is that these 3 bones are going to be very important for amplifying sound as it travels into the inner ear and the cochlea. The cochlea is full of fluid, and the outer ear and the ear canal is full of air. And so I kind of mentioned earlier that when you're playing a game like Marco Polo, if you hear somebody yelling outside of the water, so they're basically projecting that sound into air, and you duck your head underwater, you're probably not going to be able to hear it very well. And that's because when sound switches from a medium like air, where the molecules travel very freely and aren't closely packed together, and then it travels into water or fluid, where the molecules are more tightly contained, more packed together, that sound is going to lose its energy. And so these 3 ossicles basically exist to help the transfer of sound from air into fluid without losing its energy or its quality. So we have these bones to amplify sound. We're transferring from air into fluid. Additionally, one of the other things that these bones do is work to muffle louder sounds. So the 2 major muscles that are attached to these bones, both the tympani muscle and the stapedius muscle, actually decrease their vibrations when they're tensed, which allows these louder noises to be muffled somewhat as they travel towards the cochlea. So now we're going to move on to the inner ear. So the inner ear is made up of this bony structure called the cochlea. The cochlea is filled with fluid. As I mentioned, there are 3 different chambers that you can actually see inside of the cochlea if we kind of cut it apart. So right here at the oval window, that is where the ossicles come into contact with the cochlea. And then we have the smaller round window. So the area where the oval window is what we would call the base of the cochlea. The center of the cochlea is located right here. We either call that the apex or the helicotrema. You will also notice that our vestibular organs, our semicircular canals, are located here to help us with angular rotation and movement in space. Those connect to the vestibular nerve, and you can actually see here that the cochlea attaches to the auditory nerve, which will then travel to the cranial nerves and then later the brain. So the purpose of the inner ear is to basically take those changes in sound pressure and turn that into a signal that the brain can actually use. So you can kind of think of the cochlea for hearing as the retina of the eye is for vision. As I mentioned, the main component is that cochlea. It is a spiral structure that contains an an area called the organ of Corti. We'll talk more about what the organ of Corti is. It's filled with fluid in 3 different canals. So the 3 different canals are as follows. We have the vestibular canal. If you've had me for biopsych, you know this as the scala vestibuli. This extends from the base of the cochlea, so it starts at the oval window and it goes all the way to the helicotrema. This canal is the closest to the ossicles, and because it's closer to the ossicles, the pressure waves are going to move the fluid in this canal first. Then we have the tympanic canal, or the scala tympani. So this starts at the helicotrema and it extends all the way to the round window at the cochlear base. So once sound travels through the vestibular canal, it is then going to move to the tympanic canal. Now, in the middle of these 2 different canals, we have what is called the middle canal, or the scala media. This is between the 2 other canals, and it contains the cochlear partition, or the organ of Corti. So there are a few different membranes that actually separate these different canals. So the first one you can actually see here is the Reisner's membrane. This separates the vestibular canal, that top canal, from the middle canal. And then we have what is called our basilar membrane. That separates the tympanic canal from the middle canal. It is also the base of the cochlear partition. And one of the things that we're going to find is that as fluid moves through these canals, the basilar membrane will also move. And this is going to become very, very critical for actually being able to hear. So this little middle portion here, that is our organ of Corti. As you can kind of see, we have some hair cells here. We'll talk more about those soon. It is topped with the tectorial membrane, and it makes contact with what are known as spiral ganglion, which will eventually meet up with the auditory nerve. So what happens when sound enters the cochlea? So vibrations from those ossicles, so remember that the eardrum is vibrating, that's going to push the ossicles and help the ossicles vibrate and pump like a piston. Those ossicles are going to push against the oval window, and because of that, the oval window will push in and out of the vestibular membrane, and that will cause the fluid in the cochlea to move like an ocean wave. Now, your question might be, what happens if sound is really, really loud? What we will tend to find is that for particularly loud sounds, if there is any remaining pressure left over from that sound wave, it is going to be sent to the helicotrema and back to the base through that lower level tympanic canal. And it's going to be absorbed through the round window. So here's what that kind of looks like. So here is our stapes, part of our 3 different ossicles. So the eardrum's going to vibrate. Our ossicles are going to pump like a piston. That stapes is attached to a membrane that contains the oval window that will push against the oval window. That causes kind of the fluid to move. So it starts at the base. So here's our vestibular canal. We reach our helicotrema. So now we're in the tympanic canal. And that fluid is going to push out against the round window. Now we need to have a round window in addition to our oval window because the fluid needs to be able to move freely. If we only had one window, the fluid would not be able to move, and as such, we would not be able to perceive sound. So now let's talk about the organ of Corti, which is located in the scala media. This is a structure that sits on top of the basilar membrane that separates the middle canal from the tympanic canal. And as you can kind of see, the organ of Corti is composed of hair cells. So we have our inner hair cells and we have our outer hair cells, as well as the dendrites of the auditory nerve. And that's what's basically going on here. These hair cells are basically functioning as dendrites. And you can see that they are making contact with that auditory nerve. So the organ of Corti basically translates the movements of the basilar membrane in particular, and this cochlear partition into a neural signal. So the hair cells are the major sensory receptors of the auditory system. So you can kind of see what's going on here. So as the fluid in the cochlea moves, this is going to cause the basilar membrane to move. Now you'll notice that our hair cells have their stereocilia embedded in this gel-like rigid tectorial membrane. And so what's going to happen is as these move, that is going to cause the stereocilia of these hair cells to basically twist and bend and shear. This mechanical movement will be translated into a neural signal that can be sent to the auditory nerve. Typically, what we're going to find is that this information is typically going to be sent to the brainstem, but we can also actually use top-down control from the brain to actually adjust how close these hair cells are to the tectorial membrane so that we can actually tune in to quieter sounds. So we can also, this area can also receive input from the brain in addition to sending input to the brain. So here's what this looks like in an actual picture. As you can see, we see the hair cells here. Obviously, if you can see the stereocilia, this means that we have removed the tectorial membrane so that you can actually see it. So the tectorial membrane is a gel-like structure that extends into the middle canal. It floats about the inner hair cells and it touches the outer hair cells. And so the vibrations will also cause the tectorial membrane to bend and move. This causes the stereocilia to bend, not bend. And because of that, neurotransmitters will be released as a result. So in this case, we get neurotransmitter release and we get the beginning of this transduction from sound into an electrical signal by mechanical action of these hair cells against the tectorial membrane. So here's what that kind of looks like. So it's really the inner hair cells that are going to be conveying this information. The outer hair cells can also be used to enhance the sounds that we hear, but by and large, the inner hair cells are our primary receptors of the auditory system. So here's what kind of happens when the basilar membrane is moving. So what will happen is that because of sound-induced vibration, the tectorial membrane will move and it will cause our stereocilia of our hair cells to bend and shear as a result. So let's talk a little bit about these stereocilia. These are extensions of hair cells that basically initiate neurotransmitter release when they're flexed or bent. So each of these is going to be connected to its neighbor with this tiny little spring-like filament called a tip link. So here's what's really interesting. So here's what's kind of happening to these hair cells at rest. So the critical fluid for stereocilia is endolymph. Now, endolymph is very, very different from the different types of cellular fluid that we've heard about for neurons before. So if we're talking about standard extracellular fluid in the visual system or in the brain, we're usually going to be talking about things like sodium. Sodium is really the critical ion for both the visual system and for most of the neurons that are in your brain. Now, what's interesting is that for our stereocilia and for our hair cells, the critical ion is potassium, not sodium. So endolymph is a fluid that's very, very similar to extracellular fluid, but it largely is made up of potassium. Now note that the hair cells, like every other type of neuron out there, is negatively charged. And so potassium will want to flow into the cell. But potassium can't flow into the cell because these tip links prevent the ion channels from being opened. So when these stereocilia are bent, the tip links stretch, and the ion channels open, allowing potassium to enter the hair cell and depolarize it. And because of that, calcium channels can open, vesicles will dock with this membrane, and that will release neurotransmitters onto the afferent fibers of the auditory nerve. So now we know how sounds are coded in general. So how do we code for very specific amplitudes or very specific frequencies? Well, for amplitude, it's actually pretty easy. For amplitude, generally what we are going to find is that louder sounds create larger waves in the cochlea, and that leads to greater shearing of the hair cells against the tectorial membrane. That leads to greater neurotransmitter release and more firing of auditory nerve fibers. Now, for frequencies, it's a little bit more complex. We have a few different mechanisms. So we have what is called place coding, we have what is called temporal coding, and we have what is referred to as volley coding or volley theory. So place coding is actually kind of interesting. Different parts of the cochlea are tuned to very specific frequencies. So what we're going to find is that generally higher frequencies tend to be coded at the base of the cochlea. That's because the base of the cochlea is very, very thin and stiff, which means that that portion of the basilar membrane is going to vibrate more to high frequencies. So I want you to kind of think about a guitar string. A guitar string, typically the highest notes on a guitar string are with the thinnest, most tightly strung string. So generally, when something is very thin and stiff, we get greater vibration for high frequencies. On the other hand, lower frequencies tend to occur at the apex or helicotrema. The basilar membrane there is very wide and floppy, and we know that based on resonant properties of sound, that oftentimes wide, floppy sort of strings vibrate the most for lower frequencies. So the frequency of a sound will be determined by the place along the cochlear partition that has the greatest amount of movement or displacement. So here you can kind of see that here. So we have some different traveling waves with different types of hertz. So what we're going to find is that those higher frequencies are going to tend to occur more at the base, and lower frequencies will cause greater displacement at the apex or the helicotrema. So we can figure out where a frequency by figuring out the portion of the cochlea that has the greatest amount of displacement of the basilar membrane. Now, to understand the other methods of coding, you have to understand a little bit more about hair cells. So hair cells do not fire action potentials. Like the photoreceptors of the eye, they respond to graded responses. More intense, louder sounds tend to have more neurotransmitter release. Only the fibers of the auditory nerve can fire action potentials. So the inner hair cells basically convey almost all of the information about sound waves to the brain. They are basically our afferent fibers. They send information about sound to the brain, and they make contact with the auditory nerve. The outer hair cells actually receive information from the brain via efferent fibers. They're basically there to help tune the cochlea, so to speak, to hone in on certain types of sounds and basically work as a feedback system. So what we will tend to find is that one of the other ways that we can do place coding, besides looking at the cochlea, is by doing place coding in the auditory nerve. So what we tend to find is that individual fibers in the auditory nerve respond to different frequencies based on their placement in the cochlea. So information from the cochlea is sent to auditory nerve fibers, and this place coding is maintained all the way up to the auditory nerve, different parts of the auditory system, all the way up to primary auditory cortex. This is what is referred to as frequency tuning. So we can use the outer hair cells to help us tune into certain types of frequencies as well, but this really only works when sounds are very faint. So here's a couple of different examples of what are called threshold tuning curves. So basically what these threshold tuning curves show us is the lowest possible intensity for the neuron to actually fire above its spontaneous rate. So you can kind of see the lowest possible frequency that you would need before it starts firing in a way that is not necessarily spontaneous. So the lowest point that you will see on each of these curves is what is referred to as that neuron's characteristic frequency. So these are all different auditory nerve fibers, and as you can kind of see, each of them seem to be tuned to a very specific frequency. So this is less than 1,000 hertz, this is a little bit more, this one's even a little bit more, and you can see that we are coding for increasingly higher frequencies with each of these 6 different fibers. So let's talk about how the outer hair cells kind of help with that. So they kind of get feedback from the brain and can actually make parts of the cochlear partition stiffer. And when they do that, that actually causes the response of the hair cells to become more sensitive, and they can be more sharply tuned to very specific frequencies. So here you can actually see the different responses with hair cells that are active. So what you're looking at here are 3 different threshold tuning curves. Now each of them is tuned to a specific frequency, but what I'd like you to pay attention to is this little dotted purple line. This is what the threshold tuning curve would look like if the outer hair cells were not providing feedback. So the outer hair cells can actually help us tune in to very particular frequencies so that we can hear specific frequencies or sounds that we need to hear. But here's kind of the catch. Place coding may not be enough. And one of the ways that we can kind of see this is with something that is known as two-tone suppression. Typically, what we are going to find is that the firing rate of one auditory nerve fiber will decline if you present another tone at the same time. And this two-tone suppression effect is enhanced if the second tone actually has a lower frequency than the first. And this can actually affect the ability for auditory nerve fibers to code for different frequencies. So another thing that we have to be aware of is what is known as rate saturation. So the question is, are auditory nerve fibers selective for frequencies at levels above their threshold? So we kind of showed with those threshold tuning curves that each auditory nerve fiber has a particular frequency that it kind of codes for. So one of the things that you're looking at here are what are called rate saturation curves or isointensity functions. So this is for one particular auditory nerve fiber that has a preferred frequency at about 2,000 hertz, a characteristic frequency. And so tones of different frequencies, as you can see here, are basically being played at varying sound pressure levels. And what you can kind of see is that when the sound is relatively quiet, you can still see that it prefers tones of around 2,000 hertz, its characteristic frequency. But if we start increasing this intensity all the way up to 80 decibels, it becomes highly responsive to a wider range of frequencies. And so because of that, it loses its ability to actually tune for different frequencies. And so this displays what is referred to as rate saturation. Basically, at this point, the neuron is firing so much, and no further stimulation will be able to increase its firing rate. So what we're going to see is that basically at some point, especially if sounds are loud, its firing rate will max out, and we're not going to be able to code for different frequencies as well as we could when sounds were quiet. So place coding is very useful, but it's insufficient. So one of the things that we can do to actually work around this rate saturation is by utilizing the fact that auditory nerve fibers all have different spontaneous firing rates. So we have what are called high spontaneous fibers, and we have what are called low spontaneous fibers. So our high spontaneous fibers tend to work best at lower levels of sound intensity, and at higher levels of sound, at higher intensities, they're much less selective, so they tend to rate saturate. However, low spontaneous fibers tend to be pretty sensitive regardless of intensity. And so you can integrate information from a variety of fibers to actually determine that frequency. And your auditory system will also look at the overall pattern of fiber firing. So the cochlea, in addition to coding for frequency, also codes for time. And this is what's known as temporal coding. Generally, neurons exhibit what is called phase locking, which you can actually see right here. They fire at one distinct point in the period of a sound wave at a given frequency. So here we have a particular sound frequency. Here is the firing rate for a given neuron in the auditory nerve, and you can see that it largely prefers to fire at the beginning of the upward swing of this particular frequency of sound. So what this tells us is that the firing pattern of an auditory nerve fiber can help carry a temporal code. So this is what temporal coding is. It's basically taking advantage of the fact that neurons will fire in the auditory nerve at a very specific period of a sound wave cycle. So the cochlea can basically code for the frequency based on the timing of the firing as it relates to a period of sound. Now, this is only really going to work for lower frequencies. For higher frequencies, the neuron cannot fire fast enough to appropriately code over time. So what you have to do instead is potentially do what is called firing a volley. So you would want to fire a volley for sounds that are basically higher than 1000 hertz, because temporal coding tends to not work very well at 1000 hertz, and it generally does not exist for frequencies above 4000 or 5000. And we know that we can hear those frequencies. So multiple neurons will code for that frequency in a volley. So each neuron in the auditory nerve fiber will fire at a distinct point in the period of the sound wave, but may not fire every period. But the brain can add all of those different things together and get that same temporal code. So here's how this works. Here's that sine wave. So neuron A is firing during particular portions. Neuron B is firing during particular portions. C, D, and E. Notice they're not firing all the time. But when you combine their responses, we get that very nice, beautiful phase locking. So I'm going to go ahead and stop here for now. I think I've kept you long enough already. We will talk a little bit more about auditory processing and hearing in the ear shortly.